right, so let's get started. I only have 3 minutes. All right. All right. You take 5. All right. So here we are for Ethan Duvall's PhD dissertation talk. All right. So first of all, Ethan hails from Washington State. So he grew up in North Bend, which is this town that's east of Seattle, right at the gateway of the Cascades. And for those of you that don't know North Bend, you might instead be more familiar with this as a location of the cult classic Twin Peaks. All right, Ethan explains a lot in this area, this place with just this beautiful nature and so forth. All right, and then he goes off to Western Washington University, where he gets his undergraduate degree, and it was right after that that he emails me. All right, so it's really going back, all right, and looking at someone's first email. All right, so he tells me he's just graduated in environmental science. What else? He's got these awards. He's doing research on dam removal in the Elwha. That was actually the largest dam removal ever done at that point. He's interested in the ecology of tropical rivers. All right, so maybe that might be of interest to me. And then he's also interested in water birds. All right, and then so let's just wind the clock forward. And Ethan gets into grad school, decides to come to Cornell, and before he takes off from Seattle and heads to Ithaca, New York, he asked me whether or not I had any advice. He really wants to get going with grad school, all right? And so I remember there was at least 2 pieces of advice, and you can decide which of these is more important. All right, the first piece of advice I said, if you're heading cross country, you can't miss the hidden jewel. All right, and I say that to everyone in the room. All right, if you haven't been to South Dakota, there really is this gem here, right? You can ask Ethan about that. And then the other piece of advice was, before you show up, you should read this book, Ecological Stoichiometry. All right, and ecological stoichiometry is really the Bible, if you will, for this whole field that looks at the importance of elemental ratios in ecological interactions. And so by the time he had arrived at Cornell, it seemed like he had immediately become this ecological stoichiometry disciple. All right. I mean, the dams he kind of leaves by the wayside. All right. He's still really interested in a couple of things. All right. One, birds. All right. So you're going to hear about birds. And then he also is still saying he's interested in tropical rivers. And what you're going to see from his talk, I mean, his talk is, it's really like 2 dissertations, all right? The closest he really gets to tropical rivers isn't this picture here on the left, but it's much more like this picture here on the right, right? And so that will become apparent as his talk goes on. All right, the other thing about going back to these letters is it'd be like the standard boilerplate stuff, that you get from somebody who's reaching out about grad school. Yeah, I'm an extremely driven individual, all right? But I didn't realize what an incredible understatement that is, right? And so just, as an example, I mean, Ethan never published a paper until 2022. Here we are in 2026, and he's now published 19 papers, of which he's the lead on 8 or 9 of these. There's 5 more that are in review or in revision. And that doesn't even count some of this coolest stuff that's in his dissertation that isn't published yet. All right, another thing is, as he's cranking out papers, he also starts a nonprofit, right, around Amazon conservation of biodiversity and people, right? He is a Semlitz Fellow, so in the first class. So this is this Atkinson Center program fellowship in which they bring together MBA students and PhD students that are interested in sustainability. So again, he was one of the inaugural fellows. He's been a very dedicated mentor for undergraduates. So there's this pile of undergrads that he is really mentored for their honors thesis, and then he's helped them get their papers across the finish line into some really amazing publications. And then he's also been a dedicated teacher, all right? And so this is him giving a lecture at a field station in the Amazon. He's TA'd a bunch, and then he's also was the instructor of record for a writing, first-year writing course here at Cornell. All right. Ethan also pointed out that he's always exploring different avenues, right? And so this is in all kinds of different ways, and I'll just let that slide speak for itself. And I mean, he's just curious about all kinds of things, all right? And then just I want to end with this picture I took at Tipitini, so it's in the estuary in the Amazon, all right? And just, you know, again, I really just appreciate Ethan's, I mean, his energy, you know, this can-do personality. He's also just a super positive person. So with that, I'll turn it over to you, Ethan.
All right, you guys hear me? I gotta just give me 1 second, click out of this, click open. Okay, thank you so much, Alex. That was a great introduction. And thank you all for being here. This is a great group. I'm super excited to share with you today what I've been doing for the last 6 years. The title of my talk is Animals as Mobile Agents in Terrestrial Biogeochemistry. I'm going to start with the assumption that you all know that animals exist, that they all know that animals can move, and I'm going to do a little precursor on what I mean by agents in biogeochemistry to start. Now, I'll start with a basic example of a tree, a non-animal. When I walk around outside and I see a massive tree, sometimes I think, how freaking cool is it that this tree has sprouted out of the ground by sequestering carbon out of the atmosphere, by uptaking things like nitrogen and phosphorus in the soils. And so this tree has essentially evolved over millions of years to exploit the elemental building blocks that are around it. And yet when this tree dies or loses its leaves, it'll return those nutrients back to its local environment, effectively changing the chemical arena by which these ecological and evolutionary processes are going to play out into the future. And this is really what I mean when I refer to biogeochemical cycling, these interactions and feedbacks between the biological and the chemical. And so with this understanding, we can go back through deeper time and look at how key evolutionary innovations have essentially shaped Earth's biogeochemistry. From the rise of cyanobacteria that began pumping oxygen into our atmosphere, paving the way for more complex multicellular life, to the rise of terrestrial plants, which on land started to create fertile and organic soils that support so much of the life today. And eventually, we get to the rise of terrestrial animals, from the earliest dinosaurs to the amazing plethora of animals we have today. And we can ask this exact same question. How did the arrival of animals fundamentally change the biogeochemistry of our planet? Now, one way of looking at this is simply asking the question, how many animals are there? And what this square is representing is all the living biomass on Earth. I want you in your mind to take a guess what proportion of this is animals. It's a very small fraction, right? And maybe when you think about animals, you think about birds and mammals, at least I do, and that'll be much of the focus of my topic today. And that's even a smaller proportion, right? So it's a very, very small proportion. So in this theoretical battle between to be the most biogeochemically relevant organism, animals have a very tall task going up against things like plants and bacteria. And as you might imagine, part of my talk is trying to convince you that they can indeed outperform, per se, their biomass alone. They can do this in several ways. So first of all, animals can indirectly regulate biogeochemical processes. So through things like seed dispersal or pollination, they can promote the processes that are mediated by plants, but they can also suppress those through things like herbivory or soil trampling and so forth. Animals can also, oops, sorry about that. I just clicked this here. Animals can also concentrate nutrients in their bodies, right? That can become unique kind of chemical forms in ecosystems. So for example, we as vertebrates sequester massive amounts of phosphorus because we have big bony skeletons. And those can become really important phosphorus pools. Animals also have high metabolism, so they're constantly consuming material and releasing material. You can think of them, and I want you to think of them almost as nutrient processing machines as they're going, right? Their metabolisms are turning over nutrients, and they can concentrate those nutrients in their waste. And lastly, as my title kind of suggests, there's a really important distinction with animals, which is that they can move. And because of that, they become really important vectors of nutrients in ecosystems. While our tree might lose some of those leaves, some might flow down river, some might fly in the wind. Those are trivial distances compared to the locomotion of animals. And animals can move against those gradients, right? They can swim against the streams, they can move against the wind. And as you can imagine, almost all of these roles are fundamentally underpinned by a diversity of animal traits, things like body size, diet, metabolism. And I think that's what really excites the imagination. When you look across the animal kingdom, you can begin to wonder, what is the diversity of these roles? I mean, you can see the diversity that exists, but what does that look like biogeochemically? And that's really what excites my research. It really motivates my interests. And I can distill that down into these three questions. The first of which is, how do animal traits shape their biogeochemical roles? To what extent are these roles biogeochemically relevant? And can these roles influence the functioning of ecosystems? These three questions are going to be kind of the major through line to two major topics or examples that I'll give you today, which have been the focus of my dissertation. The first of which is going to be about the role that birds specifically can play in the cycling and transport of key nutrients like nitrogen and phosphorus at different spatial scales. And the second is going to be about the unique relationship between mammals and sodium, particularly with herbivorous mammals. And again, I'm going to walk you through how a fundamental understanding of their organismal biology and ecology can shape our understanding of their biogeochemical impacts and what those impacts actually mean for ecosystems. And I'll conclude by touching on just a few of the ways that humans can fundamentally alter these processes. All right. So to start, we're going to talk about birds. I love birds. They've always been part of the reason that I'm here today. But beyond that alone, there's many reasons to suggest that birds have a high capacity to alter biogeochemical processes. To start, they're found everywhere on Earth. There's about 11,000 plus species that can be found globally, so they're super diverse, the most diverse terrestrial vertebrate. They possess exceptionally high metabolic rates, so even compared to other endothermic vertebrates, they consume about 30% more food on a daily basis compared to similar sized mammals. They consume comparatively nutrient-rich diets. So you can see, while low-quality plant foliage is a common food item for many mammals, it's rarely a common item for birds. There's not a lot of birds eating foliage. Birds also grow really fast in their early life stage, but they can die pretty stochastically throughout the rest of their life. And we'll come back to that in terms of why that's important in their ability to transport nutrients in their bodies. And lastly, if you're new to the topic of birds, birds can fly. They have wings, and that allows them to be incredible vectors of nutrients. This figure is simply showing you that as birds are moving around constantly within ecosystems, they can represent these important nutrient linkages between freshwater, marine, and terrestrial systems, as well as anthropogenic systems, right? So despite all this information I've given you that really sets up birds as a high potential organism, especially among animals, to influence biogeochemistry, this research remains relatively small compared to other taxa, such as fishes in aquatic systems and mammals on land, at least from a broader and synthetic and predictive understanding. But even within birds, the broader recognition of their ability to transport nutrients remains really limited compared to our recognition of these other functions like seed dispersal, pollination. You can see transport of nutrients here is just a small sliver on this one plot. And this can really barrel down to two fundamental knowledge gaps that we have about birds. The first is which we know almost nothing about how they vary in their nutrient release, or at least our ability to accurately predict that. We also know very little about how they can transport nutrients at broad scales in their bodies through things like migration. And so these are two fundamental gaps I became really interested in from the start of my PhD. But both of these are linked to an even bigger gap, which is we know almost nothing about what a bird is actually made out of chemically. And this becomes really important because we know from previous literature on different taxa that what you are made of can influence how you release nutrients. If you're assimilating more of a certain nutrient, right, you may release less as you're growing. We also fundamentally need this information to know how birds are redepositing nutrients in their bodies when they die and how that might differ across species. So this was a starting point for my PhD and something I was really interested in. And we did a big analysis on the elemental composition of about 64 individual birds, spanning everything from a hummingbird up to a duck, a hawk, a grouse. We looked at individual tissues across birds. And though I'm not going to explain the methodologies for all of this, I will share that I was prized the most alarming process for acquiring stoichiometry data at the Conference of Biological Stoichiometry. That hangs in my office, and I'm very proud of it. Okay, so again, I mentioned that what we're made out of can influence what we release. Previous research on fishes, for example, or aquatic vertebrates, has shown that fishes with higher body phosphorus content release less into their local environment. And that's because, again, as you sequester that into your body, you hold on to that, and you're releasing less. So we really care about how species differ in what they're made out of. But with our birds, the first thing that popped out is we didn't find any differences across taxonomic identity or phylogenetic affinity. And again, this was surprising. We did everything from hummingbirds to large birds, but we started to understand this pretty quickly. The first of which is, although fish show this large variation in their body phosphorus content linked to variation in their skeletal composition, birds exhibit a relatively low and constrained body phosphorus content. And this, we think, really comes down to the fact that because birds evolved to fly, that's a fundamental attribute to 99% of birds, they've evolved to minimize their skeleton, so it's an engineering constraint, but they also need feathers, which represent about 25% of their body mass. And we found that, as expected, bone is the primary reservoir of phosphorus, whereas feathers have almost none, right? So both of these things tied to the evolution of flight have fundamentally constrained their phosphorus content. However, not across species, but within species, we do see a pretty large amount of variation. For example, body mass on the x-axis here seems to positively correlate with about a doubling of carbon content, a 50% increase in nitrogen content, whereas, as I've explained, phosphorus is relatively fixed. And this makes sense when we understand bird ecology. Birds can nearly double their body mass in fat accumulation alone throughout parts of the year. So pre-migration or before cold winters here in Ithaca, they can nearly, again, double their body mass by just fat. And you can readily observe this on birds, right? They're storing this yellowish fat in their abdominal and furcular regions. And so when we looked at this across our birds, we found that indeed, with more fat in a given bird, you generally increase the carbon content, decrease the proportions of things like nitrogen and phosphorus, but we actually found that the nitrogen to phosphorus ratio increases. And that's basically because the skeleton phosphorus is fixed, but when birds accumulate this increased mass, they may also increase some proportional amount of muscle. And again, we have our tissue measurements to confirm this. Fat is the highest carbon tissue, whereas muscle and feathers are the highest, most nitrogen-rich tissues. So once again, we now have this information that can potentially inform our predictions about how species vary and the way that they release nutrients. And so we can come up with these two basic hypotheses. Because across species, there's little variation, we might expect that it's not how you differ in your body composition, it might be differing in what you eat that's controlling what comes out, right? It's a mass balance equation. But in this mass balance equation, we know now we have to account for this temporal variation in body composition. If you're sequestering massive amounts of carbon during parts of the year, you might be releasing less, and the nutrients in your waste might be more concentrated. And what was really cool is that we were able to go out and test this exactly. So we did a massive sampling of songbirds in upstate New York, and this work led by Lindsay Chen, who was an undergrad that I was fortunate to work with, showed exactly that interactions between diet, phenological swings, and body size linked to fat accumulation together are the best explainers of how species or individuals vary in their nutrient release. Now, what about at broader scales? we know that birds undergo these renowned and incredible migrations. This little Wilson's warbler, this cute warbler up there, flies thousands of kilometers up to temperate zones to breed, but it'll fly back down in the winters to Mexico and Central America to spend its vacations. And what's really cool about this is that with this fundamental understanding that birds grow really fast, but die more stochastically, as if maybe you remember, I presented earlier, we can assume that almost all of the nutrient uptake in these birds' bodies is occurring where they're breeding, which is shown in the red here. But we can also assume that where birds are dying can occur almost anywhere throughout their life cycle, right? And in fact, these long-distance migratory journeys are when most birds have the highest risk of mortality, because it's a huge adaptive kind of trade-off between mortality and the warm summer vacations in Mexico. We know also that birds are leaving their carcasses behind. We've used things like weather radar to essentially beam birds out of the sky and show that more birds are leaving the US when they're going down to winter than are returning. But the question remains is like, what does this actually mean in terms of nutrients, and where are these birds actually ending up? So to quantify this, we integrated extensive data across about 507 bird species in North America. This includes species abundances, their full annual distributions informed by eBird data here at Cornell, the largest citizen science database. We used information about their demographics, so for any species, how many die in a year, and at what parts of the year do they tend to die? And we now have this key to unlocking, turning these mortality estimates into actual nutrients, right? We can predict the body nutrient content of a bird. And when we put these through a mortality simulation, we can simulate the uptake of nutrients into these birds' bodies during those rapid growth periods, simulate that mortality at 52 weeks out of the year, and then calculate the net deposition across species at about a 27-kilometer resolution. And so this gives us an estimate that about 13.6 billion birds out of these 507 species take flight at some point throughout the year. And among those, about half, about 6.6 billion birds may die. To give you some perspective on what that actually looks like, that's about 50% of all the savanna elephant biomass falling out of the sky, or about 100% of the New Zealanders falling out of the sky. In terms of nutrients, we know this is about a magnitude higher than recent estimates that we've derived for the transport of nutrients by baleen whales at similar spatial extents. It's not a one-to-one comparison, but it certainly indicates that this is a substantial magnitude. But where do these birds actually end up? So what I'm showing you here is a map of the net fluxes driven by birds. And what it's really showing is that in blue, birds are driving a net uptake and export of nutrients out of these high temperate zones where they tend to breed, but they're redepositing these nutrients further south throughout the United States, down into Central America, and then to a smaller extent into South America. And we see also that the highest rates of deposition tend to be along these migratory flyways, where you essentially have a funneling effect of both bird densities, but also higher mortality rates. So that's where you're seeing the highest amounts of red on this plot. And really, this cumulative effect is really something that emerges from interesting taxonomic differences. So among songbirds, for example, you have the biggest spatial extent in terms of where they breed, but also where they migrate. So they are tending to make the farthest journeys further south. Water birds, things like ducks, also undergo massive migrations, but they tend to not travel further south than the US-Mexico border. And in the winters, they tend to aggregate in massive numbers. So they create these more concentrated nutrient hotspots. Landfowl, things like turkey, are about 35% of all the biomass that we're talking about. So it's a massive amount of biomass, but their lack of large migratory strategies hinders their ability to transport nutrients at broad scales. So we know where birds are ending up. We know how many there are, but a fundamental question is how much are they actually contributing to ecosystems? And one way to get at this is to compare birds to other prominent sources of nutrients that we generally think of as the primary mechanisms of nutrient redistribution. So this includes things like dust, bioparticles like spores and pollen, fossil fuels and other anthropogenic emissions. And as a quick spoiler, when we look at this with nitrogen, we find that birds are contributing basically a negligible amount. This is partly driven by the massive quantities of nitrogen that people put into the atmosphere today. But when we look at phosphorus, we see this really interesting, these really interesting results. So when we first look at just gross deposition, in any system, how much of birds or any other nutrients are coming in, and just looking at that, we see that birds across North America actually rank third among these other sources, just behind dust and just behind biogenic particles. But when we look at the net fluxes, so in any given system, how much of something is coming in, but also how much of that same source is leaving, we see that birds are even more important, about 2 to 5 times more important than these atmospheric fluxes in North America. And the reason for this is because in any system, you might have some dust or pollen that comes in, you might have some that leaves, right? There might be some cancellation there, but birds are driving this very directional movement of nutrients from north to south, where they're uptaking nutrients is highly decoupled from places that they're depositing those nutrients. Just, I want to quickly highlight as well, we are able to validate this by comparing our estimates to that of the weather radar that I mentioned earlier, which estimated that about 1.2 billion birds die south of the US-Mexico border on an annual basis. And we find a remarkable convergence between these estimates, which gives us some confidence that the numbers we're talking about are at least in the right ballpark. Okay, so I've talked about how birds are recycling nutrients. They're these effective nutrient processing machines. They're effective nutrient vectors. They can do that at local scales, at broad scales, but what does this actually mean for their ecosystem impacts? Well, to start, we did a systematic literature review looking at about 365 studies across the last century and found that consistently where the effect of birds is tested, they tend to increase, going right of 0 here, the amount of carbon, nitrogen, phosphorus in the soils that they inhabit or the local aquatic environments that they're nearby. They dramatically change the physiochemical properties, things like pH. And this can cascade through a dynamic array of ecosystem functions. Where you have fertilization from birds, you tend to have increases in primary productivity. You are fueling plant growth, and you tend to also fuel higher plant nutrition. The concentration of nitrogen and phosphorus in plants will go up. This also appears to have many cascading responses on upper trophic levels. So you also see increases in vertebrate abundance, vertebrate body size, invertebrate abundance, and the community composition tends to consistently and almost always change when you have these nutrient subsidies that enter the system. So this is pretty cool to see. How about this broader scale? So what you're looking at here is where we know that, I guess we are estimating that nitrogen, phosphorus is being deposited in a net positive form, right? So birds are delivering phosphorus to these southern areas. And one way of estimating how important they might be is we can compare this to another map of where we've measured phosphorus limitation to occur. So where do we see primary productivity responses when we fertilize with phosphorus? And as you might notice, there's a really cool convergence between these maps, right? So this bivariate plot is showing in green all the areas where you have both high phosphorus deposition by birds, but also high phosphorus limitation. And that includes the southern part of the US going down into Central America, and then parts of the Andean foreland basin in the Amazon. And so birds are not just moving nutrients in their bodies at these broad scales, they're actually transferring phosphorus from these high phosphorus temperate zones to these more phosphorus poor subtropical and tropical areas. Now, how can humans diminish or change these processes? I could give an entire lecture just on how humans are altering these bird-mediated biogeochemical processes. Just to highlight a few of the interesting changes we've measured, we see about an 11% increase in the amount of phosphorus that birds are depositing since the 1970s. And this comes as the amount of songbirds has actually declined by about 30%, but the amount of landfowl and waterbirds has increased massively. So we have an overall increase in both biomass and phosphorus deposition by birds in total. However, if we look back prior to industrialization, we estimate that the loss of a single species, the once abundant passenger pigeon, could have transported as much or more nutrients as all of the birds today combined just in the eastern US. So clearly, we need to look back further than 1970 if we really want to get at how humans have potentially diminished these roles. But even with the changes that we've observed since 1970, it's not all sunshine and rainbows with increased phosphorus deposition, because the loss of many of these migratory songbirds has actually reduced phosphorus transport south of the US by about 11%. And we see about a 15% increase in the amount of phosphorus delivered within the US in the last 50 years. So in a sense, it almost feels like the US is stealing phosphorus from these southern countries, given that the US is driving the primary mechanisms of these changes, right? Yeah, I had a joke in my head and I'm going to say it, but I could also imagine a headline that reads, "The government blames Canada for dumping bird carcasses inside of the US." Yeah, so I'll say that. All right, so I've told you about birds, right? Birds specifically, they're really important vectors and nutrient processing machines of things like nitrogen and phosphorus. And I want to give you another example from my PhD. This unique relationship between herbivorous mammals or herbivorous animals really and sodium. You'll notice I haven't talked at all about sodium. We've been talking about things like nitrogen and phosphorus, elements that we think about as key fertilizing elements that are important for both animals and plants. And sodium is unique in this regard. So we all need sodium for our functioning as animals. We need it for sodium potassium pumps. Maybe you remember that from high school. We need it for muscle functioning, neural functioning. But most plants didn't evolve to require sodium. And instead, most plants will just uptake sodium very passively relative to the amount that's around them. And what this means is that if you're an animal that's primarily eating plants, it can be common in theory for you to experience deficiencies in sodium. There's a stoichiometric or a resource mismatch that's potentially driving these deficiencies. And indeed, across these diverse ecosystems around the world, especially in the tropics, we see all sorts of wacky behaviors. These animals exhibiting what we infer to be salt-seeking responses, whether it's tapirs digging mud out of the ground or giraffes and elephants congregating around massive salt blocks in South Africa. And so I became really interested in this, not because I just love these really charismatic animals, but again, their unique relationship with this single element. Now, part of the reason this was really confusing to understand from the beginning is because the existing theory suggests that sodium intake and sodium requirements should scale with metabolic rate in that they should slightly decline with increasing body mass. We tend to see the larger animals in these systems being the ones that are going out and looking for salt. So the fact that we infer that the requirements should actually be less when you are a larger animal doesn't make any sense. But we were able to actually show through a meta-analysis of sodium requirements that the scaling of sodium requirements actually tend to be more proportional with body size. So while you have this decline in sodium intake, or less sodium intake relative to your body size when you're larger, your requirements continue to go up. And that creates this larger gap of deficiency when you get into these larger body masses. I'd love to talk more about the physiology if anyone wants to ask more about that. We also find that the sodium requirements are about double during periods of growth and reproduction for animals. And we now have these adult requirements and growth requirements that we can use to try and diagnose these sodium deficiencies in the wild. And that's exactly what we did. We went and looked at these 3 sites across the tropics where we infer that there is this salt limitation. And what we did is we combined local measurements of the sodium content in the diet and water of these species, wrapped that into a bioenergetic model so we can estimate total sodium intake. We compared that to our new estimates of what they actually require on a day-to-day basis, and we can actually estimate to what extent are they really experiencing these deficiencies. Is this really a driver of those behaviors? Now, at each of these sites, we also have information about how often each of these individual species is going and showing these behaviors. That could be at natural soil salt licks, it could be at these watering holes, or it could be these anthropogenically provisioned salt blocks in Africa. Now, what we actually find is that about half of all the species across these diverse communities seem to experience these deficiencies at their adult stages. And so that's pretty cool to actually see. It's what we infer. We see that 100% of these species experience these deficiencies in these growth and reproductive stages. And that body mass ordered or ranked on the y-axis here tends to also correlate with these higher deficiencies. So that body mass scaling mismatch seems to really truly be driving these differences by species. And when we compare these individual species estimates to their observed behaviors, we find that consistently the amount of sodium that you obtain in your diet seems to track with a less of a reliance on these mineralic spots. So again, regardless of whether it's soil, water, or salt blocks that are providing you with this amount of sodium, it seems to be the exact same response with body size and sodium deficiency. Now, I want to take a moment just to emphasize that these are not behaviors that are like rare. They're not trivial behaviors. You can watch all of these species go and consume copious amounts of clay, shoveling it into their mouths, or licking this salt block for hours on end. And out of curiosity, I'm just wondering, out of all of you, if you had the choice between those two, who would choose the clay? Who would choose the salt? Wow, yeah. So I can tell you from a sampling of both, there's not really a right answer. But as you can imagine, this carries tremendous implications for their nutrition, their behavior, their management. And we've been able to show, actually, that the provisioning of these salt blocks within game reserves in Africa can actually lift the constraints up for these larger-bodied animals. But in turn, when you have these fenced game reserves, this can artificially inflate the herbivore populations, potentially compromising the integrity of the ecosystem. And even cooler is that this parallel work led by my colleague Andrew Abraham has been able to show that natural gradients of sodium across sub-Saharan Africa can explain the reduced densities of the largest animals. And this is, again, prominent for things like giraffes, rhinos, elephants, and prominent in the most sodium-poor places, like shown here in West Africa. So we now have two parallel lines of evidence that suggest that these deficiencies are real, that they're prominent for the largest bodied mammals in each of these systems, that they can drive changes in their behavior, nutrition, and abundance. But what about the role that these animals actually play in the participation of the sodium cycles themselves? Again, they have these unique relationships with sodium. They're going to these sodium hotspots, but they're also interdependent from plants in terms of what sodium is doing. So to explore this in more detail, I've been doing a ton of my work here in South America, particularly in the Western Ecuadorian Amazon at the Tipitini Biodiversity Station. This is one of the most biodiverse places in the Amazon basin, but also thought to be one of the most biodiverse places in the entire world. And at Tipitini, we have access to this amazing array of beautiful and intact rainforest, in which over the course of my PhD, we've been sampling for almost everything we can get our hands on, looking at the sodium concentrations in water, in fruits, in plants, in soils, in feces. And we have access to all these mineralics, so we can do a really high-resolution analysis of their behavior, of their visitation rates, and also we can sample exactly the soil that they're consuming to know exactly how much they're getting from these sites. Now, the first thing that I want to emphasize is that when we measure plants here at Tipitini, these levels, about 35 parts per million, are on par with the lowest areas we measured across Africa that I showed you previously in West Africa. So these are areas, again, we've already kind of thought to be really important constraints on these large herbivores. And when we do the same kind of modeling approach to look at their deficiencies, we find that they're even more pronounced than the three communities we looked at previously. In fact, when we look at the largest animal that we have here in the Amazon, the tapir, we are estimating they're only getting about 25% of the daily sodium intake they should need. And maybe your reaction to that is exactly as mine when I did this model, which is that cannot be right? How could anything survive off of 25% of anything that it needs to survive on a day-to-day basis? I want you to keep that in your mind. So we know that these deficiencies are happening at Tipitini. We know they're using the mineralics. We've done a ton of work to parameterize the behaviors, the environment. And so we need to incorporate that somehow into a framework to look at their sodium cycling and transport roles. And the way that we did this is we built something called an individual-based model. What's important to take away from this is that we are essentially simulating a real 1 by 1 kilometer area of the rainforest. We are parameterizing that based on the actual environmental traits. So we see about 15 to 20 times higher sodium at these mineralic sites, and we're adding that to the middle of our environment. We can now add the animals on top of this environment and give them the exact traits that we have across species, but we can also give them the exact densities that we've measured at Tiputini as well. I want to emphasize that for each of these animals, we are dynamically tracking their sodium balance, right? So we're not prescribing them a requirement. This is being dynamically tracked through those periods of growth and reproduction. And importantly, we're allowing them to actually move to these mineralic sites when they hit those critical points of deficiency. When they go to these mineralics, we can allow them to consume the exact amount of soil or water that we see them take. And we can also use, again, these visitation rates to see, is our model simulation actually driving these animals to the mineralics the same way that we see them actually doing it in real life. So what I'm showing you here is the modeled mineralic visitation, how often each species is visiting the mineralic in the model. And what I'm showing you on the y-axis is the actual observed from our camera traps. And what we see is a remarkably tight relationship between the two, slightly below the 1-to-1, which means we might be overestimating slightly for the most common visitors of these mineralics. But this R squared of 0.96 is much higher than our previous deficiency links with mineralic use, suggesting this added information about movement capacity, demography, growth, seem to be really important in terms of understanding the community scale responses. I also want to emphasize that our tapir is right on this line, right? So the ability for this tapir to go to these mineralics, receive these sodium supplements, can actually allow them to offset those massive deficiencies. And again, there's a lot of really interesting physiology I'd love to talk about, but it essentially only requires about a 5% flexibility in their sodium reservoir that they can slowly deplete through time. So now that we know this model is behaving the way that we think it should be, we can use the same model to look in the background. How does the intake of sodium and its release via urine, feces, and carcasses, how does that actually contribute to the overall sodium deposition patterns that we see? What I'm showing you on the x-axis is for each of these species, these omnivores and herbivorous mammals larger than 3 kilograms, how they each contribute to total sodium output. And the first thing that stands out to me is the evenness across these different inputs. For example, the woolly monkey is a monkey that is the most abundant primate at Tiputini. It doesn't have sodium deficiency. It doesn't visit the mineralics. And it's not a surprise that it's contributing a lot. But when we look at the tapir, the least abundant animal in our system with the highest deficiencies, it's actually contribute a similar amount because it has these massive body sizes, but also it has the mineralics that it can redistribute sodium from. However, even a tapir is not giving that much sodium back to the system than a paca, which is a small rodent, about 3 kilograms. And so the paca's higher metabolism and higher abundancies also allow it to compensate for its smaller body size. We can also look at how these animals are redistributing the sodium in space. So what's shown on the x-axis is where the sodium is ending up relative to the mineralic at 0. And as you can see, and as you can also probably predict, they're delivering more sodium near these mineralic sites, given that's where we have the highest animal densities, but also those are hubs for sodium redistribution. And what's really cool is from our model estimate there, this tracks really closely with where we actually see spider monkey are depositing their feces in space, right? So we have some confirmation that this sodium is clustering in space around these other mineralic hotspots. Now, like our birds, we also care about how much are they delivering relative to these other abiotic sources, right? And so what we've done is we've estimated sodium fluxes across the landscape for things like litter fall, rainfall, through fall leaching, and also out of the system through soil leaching. And what you can see is that our 12 mammal species here are a very, very, very small fraction. And I thought about just ending the talk there and just cutting it short, but I figured, there might actually be more to look into. And indeed, what we really need to think about is the nature of these fluxes, right? Rainfall is the largest actual flux at the landscape scale, but the actual amount of sodium in rainwater is so trivial. It's in the low parts per billions. Whereas things like urine, dung, and carcasses are these extremely concentrated sources of sodium in this very sodium replete environment. So they may actually be more important at these finer scales. And one way of looking at that is showing you tapir urination event. In a single square meter of the forest where you have a tapir that urinates, you can see that it's a much more non-trivial amount relative to these other fluxes, right? And clearly that would be even larger if we narrowed the spatial scale or the temporal scale. So what do we really think is happening? Well, we know that these animals are responding to heterogeneity at the landscape scale. These mineralics represent these sodium hotspots that, well, I first wanted to mention that we've done a ton of work looking at how these hotspots actually form, which I can't talk about, but I would love to talk later about. But the fact that you have those hotspots, these animals are responding to that, they're changing their movement, they're changing their space use, but they're also redistributing sodium away from these sites. And this can in turn potentially represent important sodium hotspots at these finer scales, right? They are biologically accessible sodium hotspots. So what could this actually mean for ecosystem impacts? I preface this all in the fact that it's probably not the plants that are going to respond to this. The plants are fairly averse to sodium. So it would have to be something within the animal system, right? And it would have to be something at these finer spatial scales. So what we decided to look at was how herbivorous termites might respond to these sodium fluxes. There's previous literature that suggests termites might also be sodium limited. And what we did is we did these 2 sodium fertilization experiments where we went out, we emulated urination events from these animals, and we measured on a day-to-day basis how termite and carbon fluxes changes. But also we did another experiment where we added a single dose, maybe more realistic of a single urination event, and then came back after a month to see how many termites were there and how decomposition had changed as well. And this is what we found. So what I'm showing you on the x-axis is the days since our first treatment here, and what on the y-axis is termite abundance in log form. And you'll see relative to the control in blue, in all of our treatments, we see increased termite abundance. In fact, in the largest treatments, we see about a 28 times increase in the abundance of termites in our subplots. We actually see the exact same thing when we look at our single pulse experiment. So we add a single dose of what we emulate to be like a tapir urination event. We come back after 1 month, and the probability of finding termites is about 10 times higher than that of our no sodium treatment. When we look at the CO2 flux, we don't find any significant change relative to our control. But when we look at the methane flux, we see this massive increase about 10 days after, especially in our 2 highest treatments. And this was really cool to see because there's a strong relationship between the availability or the abundance of termites and the amount of C4 that we're measuring leaving these soils. And this really comes down to the fact that these termites are really important drivers of the carbon cycle. They are breaking down material, but they have an important gut symbiosis in their guts that help them break down this strong woody material using a process of methanogenesis that's releasing methane. And so we can infer that potentially these animal sodium subsidies might be lifting constraints for these smaller organisms like termites, driving the carbon cycle, driving decomposition. And in fact, this is also what we see. We see that with the amount of sodium that we add, after 1 month, the mass loss of cellulose tends to be significantly higher. And I just want to point out how cool it was to do these sodium treatments and observe these responses right in front of my eyes. You might regularly just lift up some leaves and find more leaves, right? But again, after about 10 days, you start seeing these mats of termites that are building tunnels, breaking down material. It was really cool. And so essentially, these mammals we think are acting as important links between diffuse sodium at the landscape scale, concentrating that in their bodies in ways that becomes biologically accessible to these important carbon engineers. So what about the human impacts in the Amazon? I told you we're working in one of these pristine areas, right? One of the most biodiverse places in the world. But the Ecuadorian Amazon is also a place that is pervasive for oil exploration. And actually, in our research, we've been able to show that the process of exploring for oil using subsurface explosives can actually cause the creation of these extreme mineralic sites that become basically real mineralics. These animals flock to these areas And we actually see the highest amount of predators on our camera traps also at this site. We only have this for one area, but oil exploration covers almost everywhere in the Ecuadorian Amazon. So, you know, it's interesting to think about how pervasive that might be. Oil is also a huge driver of defaunation in the Amazon. You create these oil roads and they become the perfect path for hunting, and it has all sorts of community changes as well. And actually in the Maxis Road, an oil road right next to Tiputini, we used densities from those areas and showed about an 85% reduction in what animals are able to contribute to these sodium cycles. We also see that this completely severs their relationship with the mineralic sites, right? So through defaunation, we could be driving a dynamic diminishing response to the sodium participation of these animals. Okay, so I hope that the next time that you have dinner and you're putting salt on your plate, you'll appreciate the challenges that these animals have to go through to get salt, but also the important roles that they're playing. And just to recap on the examples that I've given you today about animals as agents in biogeochemistry, we've talked through how a fundamental understanding of organismal biology and ecology is incredibly important for understanding their biogeochemical roles. In birds, this was understanding what they're made out of and how they can shape our predictions of how they might differ in their nutrient contributions. In mammals, it was understanding their sodium physiology, how that changes with body size, and how that shapes their behavior. We also talked a lot about how these animals actually contribute to nutrient cycles. So in birds, we found that both at local and broad scales, they are delivering nutrients in really important forms relative to other parts of the nutrient budget. In mammals, we found that they can generate these really important sodium hotspots in sodium-poor ecosystems. In each of these stories as well, we talked about how these contributions to biogeochemical cycles can drive downstream responses within the ecosystem. For example, in birds, the fertilization effect of these nutrients can drive primary productivity and an array of downstream ecological responses from that. And in mammals, these sodium inputs may be able to lift constraints on these other sodium deficient, smaller animals at the soil scale that might not have access, like these larger animals, to the more diffuse and more landscape scale heterogeneity. And I've also talked about how we as humans have a massive capacity to diminish and reconfigure these processes in more ways that we can keep track of. And if I can leave you with anything, it's that conserving these animals is worth so much more than just their intrinsic beauty that we need to preserve so future generations can appreciate them, but it's also preserving the critical functions in biogeochemistry that they provide to ensure the restoration and sustainability of ecosystems for many decades, centuries, and millennia to come. And with that, I'll thank you.